Good morning, and welcome to another lesson with Miss Washington. Today, we're going to review protein synthesis and talk about mutations. So let's practice. Protein synthesis is the process of making proteins. To synthesize means to make something. So protein synthesis means to make proteins. Protein synthesis is broken up into two parts called transcription and translation. During this process, we're able to explain how DNA and RNA code for the proteins that determine all of our traits, like hair color, eye color, and even height. So let's practice transcription. When we transcribe, we're rewriting DNA language into mRNA language, where it's just like rewriting English into Spanish. So looking at this DNA strand, this DNA triplet strand, and using our base pairing rule that A's pair with U's, G's pair with C's, and if there is a T in DNA, it pairs with an A in RNA. Using our base pairing rule, what is our mRNA strand going to be? What are the complementary bases on our mRNA copy? Right. AUG, GCG, GUU, and AUAG. That is step one. We just rewrote DNA language into mRNA language. We just made a small copy of a gene that can now leave the nucleus and head towards the ribosome for part two. So let's refresh ourselves. What was that mRNA strand? AUG, GCG, GUU, UAG. Now we have to translate and say what that gene is actually coding for. Say the protein that that gene is coding for. The ribosome is what builds this protein. So let's do this together. Let's practice using this mRNA codon wheel. Every 3 letters on an mRNA strand is one codon, and one codon codes for one amino acid. So let's do AUG. In order to use this wheel, remember, start from the inside of the wheel and work your way towards the smaller letters on the outside. Typically, your first letter will always let you know what quadrant you're going to be in. So let's do AUG. That codes for the amino acid methionine, or MET. Let's do GCG. GCG. That codes for alanine, or ALA. Let's do GUU. GUU. That codes for valine, or VAL. And let's do UAG. UAG, which codes for stop. So, congratulations. We have just made a protein. We transcribed, rewrote that DNA code into mRNA language, and then we used the mRNA language, the mRNA code, to figure out which amino acids will be brought to the ribosome so that they can link together and make a protein. Remember, all proteins have to have a start and a stop. There is only one way to start a protein, but there are 3 ways to stop. There are 3 ways to stop. We used this way. This codon coded for it to stop. Good job. But what happens if there is a mutation or a change in the mRNA sequence? What happens if there is a change? What happens if this last base changes to a U? What happens if that last base changes to a U? Let's go ahead and make our protein. So AUG, what does AUG code for? AUG codes for methionine, or MET. GCG codes for alanine, ALA. GUU codes for valine, or valine. But let's look at this last one. There was a mutation, a random change. So UAU codes for what? What does UAU code for? U codes for tyrosine. Codes for tyrosine, TYR. So no longer is the protein stopping. The ribosome just keeps making more amino acids, or keeps adding on more amino acids to this protein. This protein no longer is told to stop. It keeps going. Do you think that'll have a negative effect if the protein was supposed to stop, but it keeps going? Let's talk about it. So, mutations in your DNA result from interactions or random and spontaneous happenings within the environment. And your genes lead to changes, which then change the function of a protein or even the physical outcome of a person. All because of one little change. So at the end of this lesson, you should be able to tell me what happens when mutations occur in DNA, what kind of mutation you think is more harmful, a gene mutation or a chromosomal mutation. So mutations, what are they? Remember, your DNA must replicate or copy before cell division. DNA replicates at 3,000 bases per minute. It's a whole lot of A's, T's, G's, and C's. It's most likely that you're going to mess up if you try to type a 3,000 character text message in 1 minute. It's going to happen. Mutations happen, y'all. These mistakes or changes in DNA are called mutations. So a mistake or a change in DNA is called a mutation. Unfortunately, cells do not have autocorrect like iPhone does. So sometimes mutations don't always get caught and fixed. Sometimes the environment causes mutation. Factors in the environment that cause these mutations are called mutagens. Some examples of some harmful mutagens that may cause changes in our DNA are things like radiation, right? Sunlight, X-rays, nuclear radiation, chemicals, and some viruses and bacteria. All of these things in our environment can change our DNA. And you know that we are a well-oiled machine with everything we need, so there doesn't need to be any changes. There are two types of mutation. There are gene mutations where there's a change in the sequence of DNA bases. So the change is in a gene, in a piece of DNA. And then there are chromosomal mutations where there's a change in a piece of a chromosome or a change in a whole chromosome. Remember, chromosomes are made up of many, 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 many different DNA bases. So if there's a change in a piece of a chromosome or a change in a whole chromosome, a lot of DNA bases are affected. So let's look at gene mutations first. Let's talk about the different kinds of gene mutations. Gene mutations are like changing a letter in a sequence. So our sentence for these examples are going to be, is going to be, the fat cat ate the red bug. So the first kind of gene mutation is called a substitution or a substitute mutation. In a substitute mutation, when you substitute something, you're switching it out for the other. So you're basically changing one base to another base. Whenever you play basketball, you sub for one of your players, you're switching out. So in our sentence, a substitution mutation or a substitution would look like changing the C to an R in cat. So the fat cat ate the red bug now gets changed to the fat rat ate the red bug. So it got substituted for another base. How do you think this mutation would affect the message? Right, the sentence would make sense, but it just means something else. So let's look at it when it's actually in a DNA base. So in this example, the thymine, the T is replaced with an A. So the T is replaced with an A. And so when it gets transcribed, it ends up coding for a different amino acid. So maybe the protein still makes sense, but it just means something different or makes a different kind of protein. Again, the protein makes sense, but it may not be making what it's supposed to. It changes the whole meaning. The second kind of mutation is an insertion mutation. When you insert something, you're adding something in. You're not taking anything away, you're not switching anything out, you're just adding. So an insertion mutation is like adding a new base. So in our sentence, it's like adding an S between the E and the F of the fat. So it's like adding a base in between those two letters. Between the fat, adding an S in between. So now, the entire sentence, what do you think is going to happen to this sentence? Does this sentence make sense to you? The sfatratat, ether de bug. That makes no sense. So, after the first word, the sentence makes no more sense. An insertion mutation is called a frame shift mutation. It's called a frame shift. It's called a frame shift because we know that DNA and RNA are read in groups of 3. Always, always, always read in groups of 3. So if this S was added in, it shifts the entire rest of the bases down. And the bases are still read in groups of 3, but now, since it's been added, the groups of 3 are different. The groups of 3 are changed. They're shifted down. So this is called a frame shift because the frame in which every 3 bases that you're reading is shifted down. So let's look at an insertion mutation here in the DNA. So in this example, A new C, a cytosine, is added between the T and the C right here. Originally, the DNA was TAA, CTG, and GC. But now, since we've added that new base, the first base, the first three letters, the first three bases are still the same, but look at this. Instead of it being CTG, it's now CCT. Instead of it being CG, it's now GCG. So it shifts all the bases down and may even code for different amino acids than it was before. The last kind of mutation is called a deletion, or the last kind of gene mutation, where there's a change in the sequence of bases, is called a deletion mutation. When you delete something, you get rid of it or you remove it. So in a deletion mutation, you're taking away a base. So it's like removing the first E in the sentence. Remember, our sentence is the fat cat ate the red bug. So it's like removing the E from the first word. So now, again, you still have to read it in groups of three. Since you deleted a base, all the other bases shift up. This is another kind of frame shift mutation. This is another kind of frame shift mutation because now it shifts the frame up. Because when you took out that base, a third space still had to be filled. But now, look, how does this affect the message? The entire sentence no longer makes sense, not even the first word. So you have a tur, a ta, tet, her, and b, ugh. It makes no sense. So in a DNA mutation, in a DNA sequence, you have TAA, CTG, CG. However, if we get rid of this A, everything has to shift up because it's still read in groups of three. So now it becomes TAC, TGC, and then G. So now it's going to code for different amino acids. This could be detrimental, especially if the correct protein is supposed to be made. So, what are the effects of gene mutations on the body or on the human, on our bodies? Gene mutations change the DNA sequence. And if there is a change in the DNA sequence, guess what? There's going to be a change in the mRNA sequence. And if there's a change in the mRNA sequence, it's going to lead to a change in a different amino acid sequence. And if there's a different amino acid sequence, that usually equals a totally different protein. So it'll be a totally different expression of our traits. And sometimes that's not good. Now let's talk about chromosome mutations, right? Chromosome mutations occur during meiosis. Remember, meiosis makes those sex cells or those gametes, those sperm and eggs. And typically chromosome mutations affect whole chromosomes. So you either have an extra chromosome or a missing chromosome altogether, or pieces of chromosomes. So the chromosome is a little smaller or a little larger than normal. When chromosome mutations happen, chromosomes don't separate correctly. Remember, in metaphase, your chromosomes line up in the middle. And in anaphase, they're supposed to pull away to opposite sides of the cell equally. However, when they don't pull away to opposite sides of the cell equally, this is called non-disjunction. Non means not or does not. Junction means where things come together. And dis means to divide or break up. So essentially, what this is saying is that where things come together and they're supposed to divide, they do not. That's literally what non-disjunction means. That means that it's supposed to break apart where it's connected together. It's supposed to divide and break up where it comes together. However, it doesn't do that. So feel free to watch this video on your own time to see how non-disjunction actually happens. So sometimes Down syndrome is caused by a chromosome mutation, or excuse me, Down syndrome is caused by a chromosome mutation. It's caused by non-disjunction where the chromosomes don't separate like they're supposed to. And so people with Down syndrome have an extra 21st chromosome. It's also called trisomy 21. You're supposed to have 2 pairs of each chromosome, but when they don't separate correctly, then you end up with an extra chromosome. The effects of Down syndrome can range from mild to severe developmental disability and include common facial features, like sunken in eyes or a smaller nose. How can chromosome mutations be detected? So you know how parents or pregnant women go to the doctor and they ask to get an idea of what their baby's chromosomes look like? Well, geneticists can actually take a sample of your DNA through an amniocentesis and organize your DNA into your chromosomes into a picture called a karyotype. This picture shows if you have the right amount of chromosomes and if they're the right size. As you can see, this is a karyotype of a person with Down syndrome because they have 3 21st chromosomes instead of 2. What sex do you think this person is? If this person has an XY, what do you think their sex is, a male or a female? Right, it's a male. So this male has Down syndrome. So when are mutations actually passed on? Organisms have 2 types of cells. This is a unit 2 reachback. There are somatic cells and gametic cells, or sex cells, or germ cells. Your somatic cells or your body cells are things like skin cells, liver cells, lung cells, muscle cells, bone cells, nerve cells, all those different kinds of cells. But there are only 2 kinds of sex cells, 2 kinds of gametic cells, 2 kinds of germ cells, sperm in males and eggs in females. So if a mutation happens in a somatic cell, like a skin cell, it cannot be passed on from parent to offspring because it's not involved in making a baby. Most cancers are results of mutations in somatic cells. So skin cells, muscle cells, bone cells, most of those mutations happen in somatic cells. This is a unit 3 reachback. Remember, cancer is when cells divide uncontrollably. However, if a mutation happens in an egg cell or a sperm cell, it can be passed on from parent to offspring. These mutations can affect the survival of the offspring. It can change the trait of the offspring and potentially be fatal. However, are all mutations always bad? No. It's true that some mutations can cause genetic disorders like cancer and death, but other mutations can actually give organisms and offspring new traits that help them to survive. Sometimes mutations can make the offspring stronger and faster and smarter. Camouflage is a mutation. Giraffes having long necks is a mutation. So sometimes they're not always bad. So think about these summary questions. What are two examples of mutagens? What are two examples of things that cause mutations that come from the environment? How are mutations detected? How can geneticists determine if there are any mutations? Compare and contrast gene mutations and chromosomal mutations. What kind of mutation do you think is more harmful, a gene mutation or a chromosomal mutation? Make sure that you answer your comprehension questions and complete your mutation simulation.